Okay, well, I'd like to welcome everyone to our September 21st meeting of the IMAG-MSM Working Group of Multiscale Modeling and Viral Pandemics. Today, we have François Graner from Paris speaking to us, taking us back to the origin of the working group, which was the beginning of the COVID pandemic. As always, I need to remind you that this meeting is live streamed and recorded and available for public viewing afterwards with light editing. Tom Helikar and I are the current co-leads of the working group. Jim Sluka and Lorenzo are the other officers of the working group. We always appreciate your suggestions for improvements and complaints, ideas, volunteers as well. We have our various social media communication channels. They only work if we use them. Please do help us get the word out. We have an archive of over 100 recordings of more than 150 seminars. Those are not used as much, perhaps, as they could be. I actually spent a lot of time in the past 2 weeks editing the formats of the descriptions so that they're consistent, so that they should be more accessible. We have the IMAG-MSM wiki page, a LinkedIn group, a Facebook group, a Twitter account. Please help us build this little community, and we really appreciate that. We have the GLIMPRINT nonprofit. We really could use help populating that website and maintaining it. So if people are willing to volunteer for that, we would be most grateful. We have the MSM Viral Twitter handle. If you retweet the announcements, that would be great. Again, we have great speakers. We want to make sure that they get an audience for their seminars. Wanted to talk a little bit about some of the things that were brought up in the past 2 weeks. There are a variety of European Union calls for proposals out, which could be relevant to this community. There's all of these under EU Horizon program. There's the Single Electronic Data Interchange Area, CIDEA, which could be of interest to people interested in standards. There is accelerating the implementation of new approach methodologies, which is quite a large program with a deadline of January 2024. There is maximizing the potential of synthetic data generation in healthcare applications. This is an area we've had some talks about recently that could be of interest to the community. There is next generation scientific instrumentation tools, methods, and advanced digital solutions. This doesn't necessarily sound like it is multi-scale modeling, but if you read the RFA, there's a big multi-scale modeling component of that with a deadline of March 2024. And there's also the Innovative Health Initiative, which includes a substantial modeling component. So for our European colleagues, of course, this is directly relevant. Many of our American colleagues might be able to find European partners for some of these initiatives. There also are 2 open calls in the United States. NSF has Ecology and Evolution of Infectious Diseases, which takes us back to the origins. Of course, we've had a number of talks on various aspects of this, zoonotics, infections and dynamics in animals, and testing of mathematical and computational models is a particular focus area of this. The deadline is pretty soon, but it's something that might be of interest to this community. There's also NSF 23614, Smart Health and Biomedical Research in the era of artificial intelligence. Again, November deadline. This could be of interest to our community as well. We will have a steering committee meeting this week, tomorrow at 11 a.m. Eastern Daylight Time. Then every 2 weeks going forward to get the steering committees in line with the seminar series. We will be talking about the mission statement of GLIMPRINT and also organizing a first in-person annual meeting tomorrow. If you think you should be there and you don't have an invitation, please let us know. The steering committee is always glad to have new members. Coming up, we have Tongli Zhang from University of Cincinnati on October 5th, Bruce Lee for the 3rd year anniversary of our series and our organization on October 19th, November 2nd. Lisbeth Garris is going to be talking about the enormous European initiative in biomedical digital twins that she spearheaded, Edith. And then we have a speaker, industrial speaker from Sotara, talking about PBPK modeling on November 16th. There are still openings through the end of the year, and of course for next year. As always, you've heard me say this before, suggest yourself for another talk, suggest your students, your postdocs, the people you'd like to hear from, anybody you think would be of interest to our community. Okay, today we're very pleased to have François Graner, an old friend of many, many of us in this community, who is going to be talking to us about the origins of SARS-CoV-2, and I will turn over the screen to him.
Thanks a lot to James and to Lorenzo for the invitation. And yes, and for this opportunity to talk about the proximal origin of the SARS-CoV-2. So my outline will be as follows. First, I will try to introduce, and then I'll make a parenthesis to draw some lessons from the past, and then focus on the COVID-19 case, explain how I can see the investigations, and then a discussion. And I will try to leave plenty of time for questions. So my introduction, I will first explain who I am and how I got involved in that, the motivations, and how we could keep open discussion channels on this subject. So for those of you who don't know me, I'm a biophysicist from a lab called Complex Systems and Matter in CNRS and Université Cité. And on this subject, I'm supervising a postdoc, Jean-Noël Lorenzi from bioinformatics. We have 3 years of postdoc funded by the University and co-supervised by Guillaume Achaz and Virginie Courtier, who are both from evolution and genetics. The motivations, well, let's start from the initial question which arose in January 2020. Well, is the origin, I mean, the question was formulated as, is it natural, lab leak, or bioweapon? I will try to argue that it should be formulated in another way, but it was like that at that time. And the 2 reasons for that question were first the place where of the outbreak, which was the city which hosted the Wuhan Institute of Virology, and also some specific features of the virus sequence. And I will come back on that later. Now, very quickly, in February and then in March, the debate was blocked in the scientific community, first by a letter in the Lancet, and then by an article in Nature Medicine, which said that the origin was natural. And in fact, the debate was blocked until May 2021, where there was a letter also in Science asking to reopen the debate, and with a immediate consequence that Facebook decided to stop censoring these messages regarding lab leak origin. Since then, there have been several publications on the subject without any consensus. And meanwhile, during this period from early 2020 to May 2021, the debate was entirely left to non-scientists, including the collective called Drastic. And of course, it was the subject of various geopolitical interferences. One of the questions which arose at the time too was related with this origin was, should we fund more or less labs doing research on virology and bat viruses? And of course, the answer would be different if the origin was decided to be natural or lab leak. And this is a decision which is important for prevention of future pandemics. With a few colleagues, we have decided to keep open the scientific discussion and keep channels open for that discussion. So we have organized workshops and entitled Evidence-Based Investigation of SARS-CoV-2 Proximal Origin. So that's why I gave this title to my present talk, with a scientific committee coming from various fields. And we held it one afternoon per month for 2 years. And then now this year, we have turned to once every 3 months. And next section will be 26. And it will be in October 19th. We also organized a public webinar on September 14th, 2021, which is still on YouTube. And it's for that webinar that we designed this logo, which represents a COVID and surrounded by bats and by test tubes. We have also written open letters, an appeal in the Lancet, and 4 open letters to the World Health Organization, and 1 to the European Union. So what lessons should I suggest to draw from the past? I will take 3 examples: AIDS, SARS-1, and the Haiti cholera. AIDS. The outbreak was in 1981 and it still lasts. The distal origin was quickly determined to be a simian virus, but there was a long-standing question for years: what was the first human contamination, and was it related with the medical campaign? And this hypothesis was rejected by the majority of scientists and scientific journals. But there were a few hints pointing at Leopoldville, which is now Kinshasa in the Democratic Republic of Congo. And in fact, at the end of the 50s, when the outbreak became important, there was an oral polio vaccine campaign, and this vaccine was made with simian samples. And so after 20 years, in September 2000, there was a debate in the Royal Society in London, a debate which was interesting, heavily biased, as analyzed by this article I quote, and debate which gave the advantage to the rejection of this hypothesis about a medical campaign. Well, in fact, soon after that debate, the scenario was, I think, well determined. I mean, that is after more than 20 years. And in fact, the initial contamination, now it's clear from the genetics and phylogeny that there have been multiple clades, HIV, and they are very old. So the first human contaminations probably took place in the west, more west, in the forest of Africa, and much earlier than 1950s. So for that initial contamination, the majority of scientists was right. But there is a but. If we are thinking about the virulence, what created a human reservoir and the conditions for evolution and transmission so that the virus became virulent, then there is a strong role of injections, prevention and treatments of several diseases in Leopoldville. And so for that point, which is important, the majority of scientists was wrong. SARS-1, well, there could have been a source of questions too. I mean, if I remind you that the initial human contamination was in markets in November 2002, and the large-scale spreading was in Guangdong in May 2003. And there was a cover-up by Chinese authorities until April. And there have been also lab accidents resulting in human infections at least six times, but after, posterior to the outbreak. So there could have been questions about a lab leak, but in fact, the consensus was very quickly reached because epidemiology, serology, and phylogeny did agree, and animal and human tests did correlate. In fact, the virulence was obtained in a proximal reservoir, which was so where the virus evolved and became transmissible. And this reservoir was a masked palm civet, very quickly identified in May 2003. The distal animal reservoir was a bit more difficult to find. It was the horseshoe bat, but at that time, the bat viruses were not well known. It took 2 years to identify that and 14 years to get really the full picture right. But of course, that was the start of a big research on bat viruses. Haiti cholera. Well, the outbreak was in 2010, and the time sequence was rather clear. For during centuries of records, there has been no cholera in Haiti. Then in 2010 January, there has been a big Haiti earthquake, and completely unrelatedly, in summer, there has been cholera in Nepal. The relation between both is that because of the earthquake, the United Nations sent blue helmets, so soldiers, and Nepal sent some soldiers to Haiti. And it was after the cholera outbreak. And a few days after the arrival, on October 14th, there was a first case of cholera below the United Nations camp. And if you see on the right, I hope you can see my mouse. Here is the camp of the United Nations at less than 1 kilometer. This is a tiny village with 15 houses, and the first case of cholera was recorded here. So you could tell that it's a very clear case. And in fact, the scientists which made an inquiry in the next month had very quickly all the scientific confirmations they wanted. The genetics, for instance, the strain was identified as coming from Asia and the same as the one from Nepal, and not like those from Latin America. Also, the epidemiological, everything was consistent, but some scientists, some leading scientific journals, the World Health Organization and the United Nations kept the idea that it was an endemic cholera, that the outbreak was only due to specific heat and rain conditions and probably sanitary conditions which were bad due to the earthquake. So this led to some fatalism, maybe more than $2 billion spent, but ill spent in bad prevention. And in fact, there were more than 10,000 deaths until in 2016, the United Nations officially recognized and apologized, saying they were profoundly sorry about their role in the cholera. Still, even 1 year later, in 2017, one of the leading scientists of the proponent of the endemic hypothesis went on sticking to environment causes. So if I want to take to summarize these 3 cases, a consensus was based on information regarding time and space, finding the reservoir of the virus, where it became virulent, determining the epidemic dynamics, and having agreement with the genetics and phylogeny. Some lessons is that scientists can be simultaneously both right and wrong, that even if you see a cover-up, it doesn't mean it was a research accident. If you don't find the reservoir immediately, it doesn't mean it was a research accident. And also that scientists can deny scientific evidence. Now, coming back to the focus on COVID-19 case, I would like to first list the consensus, then how I see the position of the problem, make a reminder about the sequence of the virus, and questions about its evolution. So the consensus, I think, is that the distal origin lies in horseshoe bat viruses, mainly rising from karstic caves in the tropical Southeast Asia, which covers part of China and also Laos mainly. And the spreading was in the end of 2019, and it was in the city of Wuhan. But I think the problem should be subdivided in 3 very precise questions. The geographical displacement, how and when did it occur? The initial human contamination or contaminations, where, how, and when did it occur? and the virulence. How did so where, how, and when did the virus evolve in order to be easily transmittable? And the order of these three events is unknown, so there can be many scenarios, especially due to the fact that each of these three questions can have an answer which is either related with research or not, and both are very plausible. So regarding the sequence, it has two key features I want to stress. The first, that it has a high affinity to humans due to its spiked receptor binding domain. Especially it has an affinity to human ACE2 receptors. And also due to its furin cleavage site, it has a very strong virulence. And to make it more concrete, the SARS-1, which didn't have this furin cleavage site, had a self-amplifying factor of around 1, so not much amplification, and it resulted in less than 10,000 cases worldwide, while the SARS-2 with it has a very strong self-amplifying factor, resulting in exponential growth at the beginning, and the figures are in multiples of 10 to the 8, hundreds of millions cases. Looking at the sequence, if we look at the phylogeny of more or less closely related viruses, we see that some regions are very conserved, or more or less, at least that alignments are correct, and that FCS is one of the cases where there is an insertion that doesn't exist in the neighbors. So if I zoom on it, you see that, in fact, it's 4 amino acids, the 12 nucleotides, and with no equivalent in close viruses. They, of course, exist in distant viruses, but the missing link, the proximal virus, has not been found yet. Another point about this sequence is that the first lineages which were found in the Huanan market differ by 2 mutations, which is not much, but too much or too few. If it's too few to think that there has been multiple contaminations by several exchanges between an animal reservoir and human reservoirs, but it can be compatible with some exchange and or it could be compatible with a manipulation in the lab, or then some 1 or 2 mutations before transmission to the market, which would act as a super spreader. So, in fact, the sequence evolution by itself does not tell us whether the evolution has been natural or manipulated, given the fact that since 2015, genetic manipulations are seamless and don't leave obvious traces. So, how could we investigate? I will make 2 suggestions regarding the cousin sequences and regarding the offsprings, and using modeling, and also I will list some other hints. So first, regarding the cousin sequences, our approach is the following, based on phylogeny. First, to make a robust alignment, and so the alignment is crucial in what follows, so it must be really as robust and objective, that is hypothesis independent, as possible. So for that, we use both nucleotides and amino acids, and we have just submitted a method article for that purpose. And we are also developing new tools to detect recombination points based on the rate of sign and mutations, where nucleotides are changed but not amino acids. So we expect to have no evolutionary pressure. We try to see which regions have evolved at different rates, and then to detect how the virus has probably cut into pieces with different origins. Using that tools, we can try to identify in the 12 most closest viruses to identify the regions which are non-recombinated and separated by breakpoints. And so for each of these regions, we will try to find the phylogenies of the region. For that, we aim to measure the rate of other changes, that is non-synonymous mutations, insertions, and deletions, and in each region, compare with the rates expected from spontaneous evolution. We will also include 38 new closed viruses, which have just been put online in research preprint. And so the method we have is quantitative and validated, promising, I would say, but in progress, and we have no results yet, and so I won't show you any. Regarding the modeling of offsprings, that usually combines epidemiology and phylogeny, and most studies, there are many papers in the literature, many methods, usually hypothesis dependent. They will all point towards a date for the patient 0, which is around September, October, November 2019. I should say that confidence intervals do not necessarily overlap, so that it's difficult to have a precise date just by making a review of the literature. And there is, of course, the question, did initial genomes disappear? We know that this kind of retrospective phylogeny has a major drawback. It has a risk of mistakes. And if, for instance, you analyze retrospectively tuberculosis or smallpox, you will find only the date of the first patient with the most active variant today. And so the old variants have disappeared. And so you will find the initial date that you find is much, much later than the real, actual appearance of this disease. Now, I would like to add a few remarks that the animals have been tested too, and so that you can do, you would like to do some phylogeny for animals, but there has been no proven intermediate host yet, even in this, whether in wild animals or farm ones. Another thing which is surprising from the epidemiology point of view is that the virus was virulent at once when it appeared, and so there was a burst within metropolis, which is a surprising pattern for epidemiology. Also, the epicenter, which includes the Wuhan market, is also close to labs, but the viruses were manipulated in Wuhan in several labs with BSL-2 and BSL-3 safety levels, so not in the level 4 safety room of the WIV. So several labs can be involved, and there is no reason to point in particular the WIV. Other rates. One interesting point is to see that Chinese authorities have decided to test and confine humans rather than test, confine, or slaughter animals, which they did when animal reservoirs were identified or suspected. The fact that labeling is a plausible hypothesis is a fact raised by Zheng Lixi herself, the well-known scientist of the WIV, in her interview of in Scientific American in 2020, where she said it was the first hypothesis which came to her mind. And in this interview, she's pictured as collecting bad viruses with protections like gloves. And we know that, in fact, collect has a risk of infection. It's her colleague and rival from the Center of Disease Control of Wuhan, Dr. Tian, who has said that on Chinese TV. And he has added also that he himself has received bad blood and urine during collects and has decided himself to put him in quarantine for 2 weeks. And in fact, this is in contrast with the current protections which are used now to collect bad viruses. There have been documented lab leaks of COVID-19, of SARS-CoV-2, but they were posterior to the outbreak. The better documented one was in Taiwan in 2021, where the lab was fined and the researcher who was beaten twice has been fired. Note an important ingredient is that in 2018, mice with human ACE2, which were part of a collaboration between USA and China, were sent from North Carolina to WIV. And the source of this information is the grant report of this collaboration. And there was also a grant submission, which was not funded, but it can be seen in this so-called diffuse project, meant to diffuse the threat of bat-borne coronaviruses. That the intention of this collaboration was to insert human foreign cleavage site in bat coronaviruses. So, discussion. I will first explain why I think there is no conclusion, but are there lessons drawn, and what can we tell, whatever the actual origin? So, no conclusion. We are still back to this initial logo with a question mark surrounded by bats and test tubes. There is no proof in one direction or another, but there are serious questions. Why didn't we find yet proximal animal hosts if everything happened in or near a big metropolis where good scientific equipment is available? Why? How can we explain the time and space coincidences? How do we explain the sequence evolution and the fact that the virus was immediately virulent at the beginning of the outbreak? Why did the Chinese authorities decide to confine humans and not animals? And why have there been no detailed investigations yet in the labs which are near the epicenter? Is there any lesson drawn for that? I'm not sure. In 2022, a collaboration between Boston and Florida has made headlines. Scientists have made a chimera using the virulence of Omicron and the lethality of the initial Wuhan strain. And with that combined, they have tried to infect mice, and they have killed 80% of their rodents. And I think that whatever the actual origin, if you forget about the past and think about the future, we now know what we have learned for sure, that there is a recipe for self-amplifying danger. If you combine high transmission rate, like Omicron, like Wuhan strain, but mainly Omicron, if you asymptomatic transmission that both strains have, and lethality, which the initial strain had, and with which, for instance, MERS has, and we know that combining the three is a real recipe for future pandemics, even worse than the COVID-19. And we know that scientists prepare such chimeric viruses, and even the combination with MERS, which has 30% lethality, has been part of the grant proposals too. And we also know that research accidents do occur. It's not a phantasm. So if we turn to prevention, I would like to promote the idea that we need to find incentives for safety, which would be much larger than the pressure to publish. And I must admit that I have no ideas of how to make such incentives. I would like to say also that if there is a lab accident, the philosophy should be to encourage alert, and finding a lab and firing the researcher is not a good way to encourage alert, for instance. I would say that collect of viruses is a way to increase the risk, and decreasing the collect, decreasing the number of virology labs, especially downtown, decreasing the gain of function research of concerns performed in this lab, I think should be a safe suggestion. And I would like to thank the collective Drastic for input for disclosing some sources. The collective also Pièce et Main-d'œuvre, who alerted me at the beginning. And my co-workers, Virginie Courtier, Guillaume Achaz, Jean-Noël Lorenzi, and also Ferial Abdi, who made an internship with us. Thanks to all workshops organizers, speakers, and participants. And also thanks to you for your attention.
Okay, thank you very much, Francois. So the floor is open for questions. If there's an issue with unmuting, please ask the question in the chat. We've certainly had some, I think your comments about safety are interesting. I don't know. I don't know. I think the natural thing is to try to play devil's advocate, but it's actually rather hard to argue with the things that you proposed.
Well, if somebody has an idea of how to have good incentives for safety, well, in industry, you have incentive which is based on money. If an accident is a risk to become very costly, that can deter people from taking too many risks. But I don't know how to have any equivalent in the field of research.
Well, I guess the one thing that you brought up, which I would say may be problematic, is the issue about the two-edged sword of collection. Because on the one hand, it's rather important to monitor and understand what the residual viral populations are if you want to be prepared. On the other hand, as you point out, doing that collection and sampling has a risk associated with it. And so I'm not sure whether there's what your feeling is about that risk balance in that particular case. Maybe the word collection has to be defined a little more tightly.
Yes, but I know I have a friend who has been in hospital for 1.5 years because of he was collecting. I mean, it was not a virus. I think it was due to a fungus. I think because he was collecting samples in very weird regions, and he was almost killed by that. Yeah. So I think there is, yeah, the benefit risk is difficult to assay, but I would like to draw the attention, especially on research of concern, gain of function research of concern, because then the benefit, I think it's even less balanced because the benefit is very dubious, because the claim is that you will be able to not only to understand, but to prepare the next vaccine. And I think there has been no proof that this type of research has ever been able to prepare a vaccine in advance, because, well, the viruses are very highly variable and it's very difficult to prepare anything and predict anything. And the risk of this type of research is very high. And like this debate has been steered by the H5N1 flu virus manipulations by Kawaoka and Fouchier. And I think the debate should be really serious and be taken seriously. Yes.
Right. The risk is obvious to all of us. If you go into increasing the strength of the virus, is there any benefit to be derived from research into understanding and treating viruses for which you would have to have that increased viral danger? I mean, if we want to do the risk benefit, the risk is obvious. Is there some benefit that goes with that kind of research?
As I said, yeah, the benefit from collecting virus is easier to understand than the benefit from really making chimeric viruses. Of course, making chimeric viruses is a much better way than collect to make headlines in the scientific newspapers and in the main press. But in terms of actual benefits for research and for health, I'm really skeptical. I've seen no theoretical or experimental proof that it really can hold its promises.
I guess one question that comes up is flu would be an example, that many influenzas don't infect my rodents very well. And so then you have an issue with what animal model to work with. This comes up also with SARS-CoV-2, that mice and rats are not a good animal model. People will work with golden hamsters or they'll work with ferrets instead, because the infection pattern is different depending on the species. So maybe one thing to say would be, instead of trying to do a gain-of-function mutation on your virus, you should look for a better model animal to work with. I don't know if that's one way of trying to square that, sir.
Yes, but working with humanized mice means that you will work with a virus which will be potentially virulent for human. So
I don't have an answer. I mean, I just there was a question in the chat from Severine that she said that the metropolitan outbreak in SARS-CoV-2 was unusual in terms of its pattern. Could you explain what a more usual pattern would be?
Yeah, what was unusual is that the virus was immediately virulent when it was first observed, while if the evolution towards virulence means that not only the sequence should evolve, but also the conditions of transmission should evolve. And that can happen. Basically, there are two main patterns. One is that you have a reservoir where it evolves for a long time, and then you have several exchanges. Or you have a human reservoir where it evolves, and basically you have the pattern of the SARS-1 and the pattern of AIDS. Those are rather typical ones, but this really arriving at the middle of a metropolis like that, where you don't find so many exchanges with animals, and where you don't have animals interacting with each other so much, is very surprising for epidemiologists. Which is, again, not a proof of anything. It's still compatible with different hypotheses. Yeah. Yeah. Yi?
I heard or read anecdotal reports or stories that earlier in the fall of 2019, there were lots of cases of pneumonia-like symptoms in, for example, Southern California and other parts of sort of clusters of flu-like symptoms and pneumonia-like symptoms. And the hypothesis was that it could be that earlier or several earlier, less virulent strains were circulating in much smaller scale and much milder, but some of them actually accumulated and mutated, and then just perfect storm scenario happened to end up in Wuhan during the winter break when everybody was inside and everybody was gathering for the Chinese New Year.
This is one of the possible scenarios. Yes, this is, and in fact, it still is compatible with what I call the consensus, which is the distal origin in bat viruses and the spread in Wuhan, which were the super spreading events were there. I think there is a consensus on that. And as I said, there is a lot of possible scenarios, and one through the United States is not excluded. I don't see any reason to exclude that. There has been the pneumonia that you mentioned have been associated with, I would say, electronic cigarettes. I don't know how you call that, but I'm not aware that it has been really proven. So again, then it still raises for me the three questions: how did the geographical displacement occur? How did the first human contamination occur? How did the virus evolution occur? And for me, these three questions can have, even in that scenario, an answer related with research or not. And so I think it's the door is really open.
Agreed.
We had Philip Ball talking a year or two ago about some of these issues.
Yes, I know I am a bit outside of the subject of the main subject of the working group. Yeah.
But I think it's interesting to ask the question then, and you alluded to it, but you didn't go into much detail, which is there a way we could how could we use modeling to try to, you're not going to be able to answer the question, but you could at least try to develop probabilistic scenarios for some of these things.
Yeah, well, my personal answer, but which might not be the good one, I mean, my answer is that modeling modeling the sequences before the outbreak, that is the cousin sequences, and finding the ancestor and the evolution is one way, and modeling the sequences posterior to the outbreak, that is the offsprings of the initial strain, are two very completely different strategies with different goals, different methods, and different type of information that we can get. In my opinion, the literature focused more on the latter, and I'm not sure it can really give, I mean, of course, having the patient zero date is important, and tracing the displacement is an important information. It's a bit difficult to make a real proof, to hope to make a significant proof out of that. Our approach might be more linear in terms of going to the proof, but of course we don't know whether we will have a significant result. We could have a significant a result significantly compatible with the natural evolution. We could have a result significantly incompatible with natural evolution, but we might also have an intermediate result which is not significant. And when we asked for the funding, we were very clear about that point, that we launch this research without knowing whether we would have at the end any significant result. It's not up to you. What we will try to do is to have really the best and the most robust method, but we cannot guarantee that the result will be significant.
I guess, I mean, let me try to understand, because we did have a talk long time ago by people who look at mainly influenza, although they were beginning to look at SARS-CoV-2 strain divergence in the human population. And so you can ask because evolutionary prediction is always hard, because something like a recombination event is very rare and has a big effect. And so how do you account for, how do you put in a simulation the frequency of those kind of events? It's a very difficult, you're very sensitive to that. Even sort of single base substitution questions are not neutral. And so then you say, okay, now let's look at how SARS-CoV-2 has evolved in the human population. How has it diverged? And one thing you mentioned, and again, I'll get this wrong, you'll please correct me, is this issue, for example, of some of the strains perhaps evolving in Africa in immunocompromised individuals, where you could have the same virus resident in an individual for a long time, so you could accumulate a lot of mutations in an individual. That's a little bit different from trying to understand the origin of the virus, but it does give you some sense of this of a very question, which is the evolutionary potential of a virus.
No, as I said, evolution means many things, including becoming virulent. And so and virulent means not only that you can, the virulent is independent of lethality, of course. So lethality is one point. Ability to contamine and to be transmitted is something else. And of course, in the case of COVID-19, one very important point, not specific to it, is that it's asymptomatic transmission, which of course for us human makes things much worse, because then you have to suspect everybody, and so life is more difficult because of that. But I would say that evolution can be much quicker if you have a reservoir of many people. For instance, in the case of AIDS, where clearly the initial contamination, there have been several initial contaminations. Somebody has estimated that maybe one per decade, maybe for 100 years, I don't know. And in different places, but usually they didn't lead to any outbreak. What happened in Leopoldville was massive campaigns of injections and without any precaution, of course. So with the same syringe being reused several times. So then you create the condition for the virus to be able to evolve. And that's much more than staying a long time in one individual. At least for AIDS, the scenario you mentioned has not been effective. While as soon as there was a massive number of people, it made the virus much more virulent. Then the very likely scenario is that only one person went to Haiti, and then and it was enough to make the world outbreak, because in Haiti there was a massive blood transfusion system with mixed bloods, and so people were massively contaminated, and then it went to the United States and worldwide. So of course, yes, there can be, and then if you make retrospective analysis, you would find only this person, which is not the patient zero at all. Well,
right, the extinct clades you don't see. Yes,
exactly.
No, when I wasn't talking about the origin of HIV, I was talking about the concept that some of the newer strains of SARS-CoV-2 may have evolved in immunocompromised individuals.
Yeah, as I said, you need much time. I mean,
Right.
Having a lot of people contaminated is a much more efficient way than to having one individual contaminated for a long time. But of course, you're right. Both scenarios exist. Yeah.
I mean, I think one thing that was a bit of a surprise, starting from 2020, was that SARS-CoV-2 evolved faster, I think, than people expected it would. They thought, well, influenza, because of its split genome, would naturally have a lot of recombination and evolve rapidly, but that maybe because you had a unitary genome for SARS-CoV-2, the rate of evolution would be slower, and that doesn't, I don't think, seem to have been the case. I don't know. Again, you can.
This point is interesting for modelers, and I am not sure I can extract information about the origin based on that.
No, I've changed the topic a little
I agree.
bit.
I agree. It's very interesting for modelers. It's very surprising
right.
too. Yeah.
But I thought, again, the origin question brings up a lot of questions that are of importance beyond the core question of origin. In other words, many of the issues you've raised would apply to trying to understand how the epidemic will play out, what the future evolution of the disease is, how do we handle it. I mean, today, of course, in the past few weeks, we've been reading about the sporadic outbreaks of Nipah virus in India, which is, of course, a topic that was raised in this group many years ago by somebody who collects bat viruses as a particular area of concern. I mean, that's one. If it becomes if Nipah virus becomes more virulent, then it's sort of one of your worst case scenarios, right? You have a virus with a lethality of 35–40%, and if it somehow starts spreading from human to human, you're in trouble. At least if the incubation is long, right? I guess the other one that you might have used as a model, and maybe you could compare, maybe because you have so much burnout, would be.
Were you talking about Ebola?
Yes, I was going to say Ebola is one example where you get spread, but it seems to because it's so lethal, you seem to burn out. And so I guess you might be talking about Ebola versus some of the other examples might be useful. Okay, we've had our discussion. I want to thank you then, Francois. Okay. And there's a lot to think about, and we appreciate your willingness to come and educate us today. It was very kind of you.
And thanks to all of you for your attention.